Hello, and welcome to this video on eukaryotic gene regulation. I'm glad you're here. As you go about your day, do you ever think about how your body, which is thought of as a single unit, a single organism, is really a vast collection of individual cells? These cells all work together to allow you to sense and interact with your environment. For example, you can see that delicious-looking burger floating in the air, think, "Yum, I'd like to eat that," and move your hand to take it. Your body has hundreds of different specialized cell types. For instance, rod and cone cells in your retina detect light reflected from the burger. Many types of neurons and astrocytes in the brain enable you to recognize the object in front of you as a burger. Then muscle cells, or myocytes, allow you to reach out your arm and take the plate. Many types of immune cells, such as B cells, defend your body from unwelcome pathogens which might be lurking in undercooked food. All of these cells have different shapes and characteristics. They express different proteins, and they respond differently to stimuli. Yet they all, well, mostly, share the same fundamental program or set of instructions, your DNA. It may sound trite, but consider that all of the cells in your body were generated from a single fertilized egg. This fact never ceases to amaze me. How does a single cell differentiate into an entire organism? And how do cells respond to their environment? These are fundamental questions in eukaryotic gene regulation. The answer is simple, but also complex. There are many, many, many ways in which gene expression is regulated. Let's look at an overview of the main levels of gene regulation. The first thing to consider is subcellular localization, a recurring theme in gene regulation. We have a cell, and within that cell is a membrane-bound nucleus. The first layer of regulation occurs within that nucleus, at the level of the DNA and the genome, and includes things such as histone modification, regulation by certain non-coding RNAs, DNA methylation, and chromatin structure. The next level of control occurs during transcription initiation and is mediated through transcription factors, commonly abbreviated TF. Note that there are both general and specific transcription factors. We'll see an example of a specific transcription factor later on in the video. Next comes RNA processing, which includes splicing in the nucleus, export from the nucleus, and RNA degradation, which is regulated in a number of ways, including through microRNAs. In the cytosol, we have translational regulation, which includes things like microRNA-mediated ribosome stalling. Finally, once the protein is made, it can be regulated by a number of post-translational modifications, such as phosphorylation, ubiquitination, localization within the cell, and degradation, or protein turnover. Every single gene is regulated on these 5 levels, perhaps differently depending on the cell type. It can get very complex very quickly. Let's examine a hypothetical situation with 2 different cell types in the genes A, B, C, and D. Cell type 1 expresses a transcription factor which activates transcription, as shown by this small arrow, meaning it's a positive regulator. The central dogma proceeds as usual, and protein A is generated from gene A. However, in cell type 2, the promoter and binding site for the transcription factor on gene A is methylated, indicated by the little lollipop dots. The DNA methylation prevents the transcription factor from binding, and no transcript is produced. At gene B, the transcription factor can bind to the promoter, producing a primary transcript which is then processed into mRNA. For cell type 2, the mRNA is then translated and protein B is produced. But cell type 1 also expresses a microRNA. This microRNA binds to mRNA B. It could either lead to its degradation or stall its translation at the ribosome. Either way, less protein B is made in cell type 1. Now let's take a look at gene C. In cell type 1, nothing hinders the expression of the gene, and protein C is produced. However, cell 2 expresses a repressor that binds to a regulatory element upstream of gene C. This repressor inhibits the binding of the transcription factor, inhibiting transcription, meaning no mRNA is produced. Finally, we come to gene D. In cell type 1, protein D is produced, but unless it's modified further, it's inactive. In cell type 2, protein D is also expressed. In cell type 2, protein D is also expressed, but importantly, cell type 2 also expresses protein B, which activates protein D via phosphorylation. Let's take a minute to review the 5 levels of gene regulation present in this example. First, there is genome regulation, 1, at gene A. Then there is transcriptional regulation, 2, of gene C. For gene B, we had 2 potential mechanisms, either RNA processing, 3, or translational regulation, 4. Finally, we saw an example of post-translational regulation of gene D, 5. Now, how could we tell these 2 cell types apart? First, let's take a minute and make a summary table of the differences between these 2 cell types. We have genes A, B, C, and D for both cell types, and we specifically looked at 2 outputs, mRNA and protein. In cell type 1, mRNA was expressed for all genes. However, in cell type 2, there was no mRNA produced from genes A and C, but we did get mRNA from genes B and D. On the protein level, cell type 1 produced protein A and C, but not protein B, and protein D was produced, but it was inactive. For cell type 2, since no mRNA was produced for genes A and C, no protein was produced either. But both proteins B and D were made. Now, how would you determine this experimentally? One of the most common methods to study gene regulation is to quantify the relative abundance of an mRNA by qPCR or RT-PCR. For protein expression, a Western blot, abbreviated WB, is a common method. Let's start with what the qPCR results might look like. On the y-axis, we have relative gene expression, which is basically relative mRNA abundance. And on the x-axis, we have all of the genes. For cell type 1, all of the mRNAs were expressed, so I can add them all in at 1, since they're normalized to themselves. Now for cell type 2. For gene A, since we had no mRNA produced, the relative expression is much lower. For mRNA B, if we assume a ribosome stalling mechanism, the mRNA levels might be similar. For mRNA C, its expression in cell type 2 is much lower, and for mRNA D, the expression is the same, since the only difference here was a post-translational modification. But is this the whole picture? It's a good idea to run a Western blot if you have good antibodies for proteins A, B, C, and D. I'll set up a hypothetical blot, with the latter enabling me to determine protein size and kilodaltons. For protein A, you would see a lot of it in cell type 1, but very little in cell type 2. For protein B, there would be some of it in cell type 1, but much more in cell type 2. Protein C has high expression in cell type 1, and almost no detectable expression in 2. Now protein D is interesting. It would be present in cell type 1, and most likely in cell type 2 as well. But be careful. Do you know what portion of protein D the antibody recognizes? How would the results differ if the antibody didn't bind to phosphorylated protein D? Or, even better, is there an antibody that can specifically detect phosphorylated D? This example shows that investigating gene regulation on multiple levels is vital to revealing the whole story. Let's rewind a bit. How are these different regulatory mechanisms established in the first place? Why was gene A methylated in cell type 2, but not in cell type 1? At first glance, this seems like the classic question: which came first, the chicken or the egg? The regulatory element or the transcription factor? In reality, all of the complex regulatory networks can be traced back to development. As cells differentiate, the way the genome is regulated and expressed changes. The accumulation of these changes helps generate the vastly different cell types in our bodies. But how to envision this process? I like to imagine a decision tree where each split indicates a different signal that influences cell fate at that branch point. As cells progress through differentiation, they follow the path that characterizes their particular cell type. But how could a signal promote one cell fate over another? Let's erase part of this so we have a simpler diagram. Imagine that in some point during development, we have a group of cells that will go on to generate lineage A. There are a number of genes that, when they're expressed, promote cell fate A. Now, another subset of the same group of cells will go on to generate cell fate B. And there's another set of genes that promote cell fate B. So the genes in cell lineage A express the blue genes associated with A, and lineage B cells express the red genes associated with B. But how are the lineages established? There are lots of ways this might happen, but let's say that signaling molecule A is vital for establishing cell fate A. As signaling molecule A diffuses through the embryo, it only reaches a subset of the cells in question. Binding of this signaling molecule triggers a chain of events that activates transcription factor A. This transcription factor then goes on to activate the expression of the genes that promote cell fate A. In addition, transcription factor A also activates the expression of gene X, which produces protein X, a chromatin remodeling protein. Protein X binds to the genes that promote cell fate B and recruits a chromatin remodeling complex that condenses chromatin. Condensed chromatin at these loci inhibits transcription of these genes, repressing cell fate B. Since chromatin condensation is a relatively stable gene alteration, the genes that promote cell fate B may be shut off in all daughter cells of cell fate A. Now, what happens to the other cells that weren't exposed to signaling molecule A? In these cells, transcription factor A is not activated, so the A-promoting genes remain off, repressing cell fate A. In addition, no protein X is produced, meaning the B genes remain on, promoting cell fate B. Thus, these cells will differentiate into cell fate B. Understanding how cells acquire the correct fate and accompanying gene expression programs is an active area of basic and biomedical research. Now let's take a look at a real-life example of multi-level gene regulation that occurs in a single pathway: NF-kappa B, or nuclear factor kappa B, signaling in immune cells. NF-kappa B is a transcription factor that binds to a specific consensus DNA sequence to activate gene expression. These sequences can be found upstream of many genes. When the pathway is inactive, the NF-kappa B dimer is located in the cytoplasm and is bound by I-kappa B, or inhibitor of kappa B. Inhibition keeps NF-kappa B out of the nucleus. But when the cell receives the right signal, for example, the activation of the T cell or B cell receptor, the pathway is activated. The pathway has more steps than we'll discuss here, but ultimately, activation of it leads to the phosphorylation of I-kappa B. Phosphorylation then recruits an E3 ubiquitin ligase, which polyubiquitinates I-kappa B. This targets it for degradation by the 26S proteasome. This whole sequence of events releases the NF-kappa B dimer, which is then free to translocate into the nucleus via its nuclear localization signal, or NLS. There, NF-kappa B binds to its consensus sequence and promotes the transcription of genes that further activate the immune response. As you dig deeper into the pathway, there are other levels of regulation going on as well. For example, the NF-kappa B consensus sequence is located near many genes, but these sites need to be accessible for NF-kappa B to activate gene expression. For example, binding of a repressor or silencer near the NF-kappa B site may physically prevent the dimer from reaching its binding site. This is another example of regulation at the level of transcription. With NF-kappa B signaling, we also saw many elements of post-translational regulation, including protein modification, with the phosphorylation and ubiquitination, protein degradation, or protein turnover, and protein localization. Now, how would you determine if NF-kappa B signaling is active, given the pathway above? What would qPCR or a Western blot reveal? I'll conclude with some of the key concepts that we've gone over. First, I hope you have an appreciation for the beauty and complexity of eukaryotic gene regulation. I think this stuff is awesome, and I hope you do too. Okay, down to business. Gene expression is regulated in many ways, including at the levels of the genome, transcription, RNA processing, translation, and post-translation. Next, complementary methods can and should be used to investigate gene regulation at multiple levels. There's another video that goes into more details on the various methods used to study gene regulation. Next, gene regulation is cell type and tissue specific. This is how a single set of instructions, your DNA, leads to the thousands of different cell types in your body. And finally, development is orchestrated through the regulatory programs of many different cells acting in tandem. This has been Eukaryotic Gene Regulation by Emily Jobe for BioCore 383 at the University of Wisconsin-Madison. I hope you enjoyed this video, and thanks for watching.